to talk about one of my favorite creatures, the Rio Grande Siren. This is composed of numerous years of research to get to where we are in terms of the status of this species. And let's see if I can advance my slide here. So we've actually had 2 state wildlife grants funding this project. One, it started around 2013, talking about the population genetics of the threatened South Texas siren, and then this recent state wildlife grant defining the range of the South Texas siren, and they had multiple components to them. There were 2 master students that worked on these projects, and I'd like to acknowledge them because they did a lot of work and got very muddy and wet in these swampy areas looking for sirens. Taylor Dufik and Krista Rupert both contributed a lot to these studies. And I would also like to acknowledge the people that taught me about sirens and helped me understand where to find them and how to catch them down here in South Texas. One major player was Max Pons, who was previously at the Nature Conservancy. He had some in a stock pond or a little loading dock in the back of the building. And we would actually practice catching them back there. Andy Glusenkamp helped quite a bit to understand the problem down here regarding the siren. Seth Patterson, a local naturalist, really helped out in the field and really knew a lot about the area. And Clint Guadiana at the Gladys Porter Zoo also has been a big help in our eDNA project. There are numerous other people to thank. Pablo Quintanilla at Sabal Palm Sanctuary, Melissa Jones at TPWD, and then we've had numerous students and volunteers helping. Of course, Texas Parks and Wildlife, the Nature Conservancy, Gladys Porter Zoo, U.S. Fish and Wildlife. Thanks to all y'all. So a little bit about the siren. They're aquatic salamanders. They have an elongated body and eel-shaped external gills and reduced forelimbs, and they're often confused for eels. They're found only in North America and in the southeastern United States and northern Mexico specifically. They inhabit permanent or ephemeral water bodies, and they have some unique attributes such as being able to estivate. They can make a mucus cocoon and stay underground when times are dry, and they can stay underground for 2 years or more if they're large enough. They can make up an enormous portion of the biomass of water bodies that they inhabit. For instance, in Florida, there can be up to 1.3 sirens per meter squared in some of these ephemeral areas. So therefore, they play a key role in wetland habitats and predator-prey relationships. And they occur in lots of these ephemeral water bodies, and they end up being good biological indicators as well. In terms of their habitat and physiology, they're adapted to hypoxic and anoxic environments. They have both gills and lungs. However, in our observations, they really seem dependent on aerial respiration, breathing from their lungs. They use the gills as an accessory, maybe to stay down a little longer, but they can drown if they have no direct access to the surface, kind of an Achilles heel, so to speak. Here's some long-term oxygen data that we collected at Sabal Palm Sanctuary, one of our ideal locations for collecting sirens. And as you can see, this area goes hypoxic pretty much every day. And I think that gives the sirens a little advantage. Some of the larger fish predators probably can't survive in this area unless they can breathe air. And they can be the dominant predator species in some of these areas. So sirens disperse across the landscape as adults using sheet flow. So we get these torrential rains at certain times of the year due to hurricanes and tropical storms. And these animals just pick up and walk across the landscape. They're not really very good at walking at all. They have 2 forelimbs, no hind limbs, and their gills kind of drag along the ground while they're walking, but somehow they do it. And we've had many people call us and say, hey, I saw this weird eel walking down a dirt road. What is it? And they get across the landscape and disperse into new ponds by that method. I'm sure they also disperse as larvae when ponds overflow, but it seems like these kind of long-lived species can disperse across the landscape as adults much easier. So some of the potential threats to the siren are habitat being degraded and fragmented due to numerous reasons, some of them being the dredging. These oxbow lakes we have down here kind of fill up with sediment, and they want to dig them back out for water control. Sirens have been seen during the dredging events being dredged up. Sand mining, there have been numerous reports of sand trucks pulling up, dumping out their load, and a dozen of these kind of rubber ball looking objects come rolling out that are actually estimating sirens. You've got agricultural runoff with pesticides and herbicides. You've got the border fence where they're building concrete footers along the whole stretch of it that would be an inhibition to their dispersal method. Urban development, bulkheading of these resakas, and even altered drainage regimes, so you've got more road runoff running into these ephemeral ponds. So the genus siren has been somewhat confused because they're hard to identify. And the South Texas siren was declared threatened in 2003 by TPWD because it didn't have a lot of information about it, and there seemed to be a large form that occurred down near the border. There are 3 extant species described now. There's a greater siren, which gets to be the size of a fire department fire hose in diameter and about 1 meter in length. The reticulated siren that has a very distinctive color pattern that we described in 2018. And then there's the lesser siren, or Siren intermedia, which appears to have 3 subspecies that were described in the 40s and 50s due to some dubious characteristics. So if you look on IUCN and Red List, you can see that Siren lacertina is described all over in Florida, Georgia, and South Carolina, and a little part of Texas for some weird reason. And then you've got Siren intermedia that kind of overlaps in some of its habitat as well. And this is where we came in. We wanted to know, well, are there really Siren lacertina down here? And which one is the large form SP1 that we're looking for? If you look at the other related literature, such as Dixon's guide, you can see that Siren intermedia texana is described as occurring in South Texas, and Siren intermedia nettingi occurring up in northern Texas. Tipton et al. In 2012 described the Rio Grande Siren species occurring in this kind of U-shaped area down in South Texas. And it's unclear what all methods they use to define this. Some of these said genetic studies ongoing. And so siren species were kind of identified first visually and by morphology and by features called costal grooves, little grooves along the side of their body that people would count to try to identify them. Here's a picture of the Rio Grande siren. Here's a picture of Siren lacertina, the greater siren, and here is Siren intermedia. And I don't know if you can tell the differences between those. It's pretty hard. But the other problem is that the sizes and the costal groove counts and even the coloration patterns overlap in their descriptions. And in fact, we did a photo ID exercise with sirens down here, and we took pictures of their heads and tried to identify them by the spotting pattern. But these are just from the Rio Grande Valley. And you can see the coloration pattern is not going to help. The head shape is slightly different, and some of these more rounded and more narrow. It's really hard to tell what you have by coloration pattern. We also gave up on trying to count costal grooves because you can miscount very easily. So what we've done is captured sirens using minnow and crayfish traps and tried to identify them genetically. And so we use floats in each trap, and we use chicken livers to capture them because that seems to be the best bait. And if you look in the center picture here, you can see a kind of a characteristic habitat for sirens. This is an area where we would catch 10 or 12 sirens at a go. So we sampled 19 counties around Texas, and we added 23 additional samples to this project in our genetic analyses. And we tried to span the area that might include lacertina, intermedia, and the 2 subspecies that people have identified in guides. One thing to mention about the siren is that it has a very large genome. Over here on the left, we have the human genome size, which is about 3.5. The sirens range from 55 to 60 in terms of their genome size, and therefore 15 times larger than the human genome, which makes them a very hard species or group to study genetically, because there's so much extra parts of the genome we don't even know anything about. And so we chose to actually analyze them using mitochondrial genome sequences. Typically, people use cytochrome oxidase 1 and 16S ribosomal sequences to identify and to publish on GenBank. These are small regions, and some of them can be misleading. So what we decided to do is actually use complete mitochondrial genome sequences to analyze this population. So initially what we did was we amplified 3 large fragments using long-range PCR, as you can see in this diagram, these gray long arrows going around the mitochondrial sequence. We put them into a plasmid and cloned them and amplified them, and then walked around the sequences with primers and then Sanger sequencing. Later studies, we used the 3 large fragments, but instead of primer walking, we actually used Illumina next generation sequencing to speed up the process. And so here's this colorful diagram is just us aligning and concatenating the sequences for the different samples. And we use 16 coding and non-coding regions of the mitochondrion. We then performed analyses and made maximum likelihood trees and Bayesian inference trees to help us understand the phylogeny of this group. And some previous work that we also did with the reticulated siren showed us that Siren intermedia down here in Texas is quite different than the Siren intermedia identified even in Louisiana and Florida and even Arkansas. And so it looks like we have some kind of a different group down here in this area. Here is the phylogeny from using Bayesian inference for the 23 samples in 19 counties that we completed in this study. We used the coding and non-coding regions, and they differed by only 1.6 to 2% in this in these sirens that cluster together here. And this ranged from an area from Cameron County, west in Kinney County, and all the way north to Tyler County, where we had an apparent monophyletic group here. There were some divergent genotypes also seen in several areas, such as Hidalgo and Cameron County, Colorado County, Houston, and even Jasper County. And it's unclear exactly what's going on there, but it looks like the majority of our samples form a monophyletic group. So in general, the mitochondrial analysis indicates that there's at least one monophyletic group in Texas. It seems to be distinct from Lacertina, the greater siren, and Siren intermedia that's in GenBank and identified from the eastern U.S. We found no evidence so far for a large form in South Texas, although we have found individuals up to about 600 millimeters. They still group with this monophyletic group. The divergent genotypes may have several explanations, such as being brought from other areas of the country. They're used as pets sometimes. They're also historically been used as bait in some areas. And we also have a lot of fish farms that were prevalent down in this area over the years. And water carrying catfish and other things may have also led to some dispersal events of sirens. So the predominant form occurring throughout Texas does not appear to warrant threatened status in Texas. That's not to say that maybe there's another large form that we couldn't capture. We did considerable amount of sampling, but I think the only way to resolve this problem may be to do eDNA metabarcoding and search for these sirens using water samples, because the trapping methods we've trapped until our heart's content, and most of these fit in that one monophyletic group. There are still many threats to wetlands and dispersal for sirens, such as roads and even the border fence construction, because of these concrete structures that may prevent them from crawling over with their only 2 legs and kind of dragging their gills on the ground. So that's something to think about in the future for these sirens. And with that, I'm going to hold questions until the end of the presentation, but Dr. Drew Davis will take over the second half of this presentation regarding the development and assessment of an environmental eDNA assay to detect Rio Grande sirens.
All right, Paul, can you hear me?
Yes, sir.
All righty. So thanks, Rick. Yeah, as Rick mentioned, we're going to sort of transition to the use of a technique to detect sirens in South Texas. Environmental DNA is this emerging, recently developed field that is based on the extraction of DNA from the environment that's used to detect species. And so this DNA is typically shed from organisms from urine, feces, and skin, and that's what's being captured and used to detect species presence. And so really the benefits of eDNA sort of rise from detection without the need to capture, see, or hear species of interest. And overall, it's sort of considered to be a less labor-intensive method, less invasive, and can be quite cost-effective depending on sort of the modes and goals of the research. However, with each eDNA assay that's developed, the methods of this assay need to be validated for the species and sort of fine-tuned for the habitats that the eDNA collection is being done in. So traditionally, sirens were surveyed across the landscape with trapping, and you know, numerous papers and have sort of shown success with different types of traps and styles, baits, and different things. However, you know, from experience at sites down here, we have sort of periods of success and periods of failure where we know sirens are at a site, but we can't seem to detect them. So you know, this eDNA assay may be a better way to detect sirens on the landscape, and but to really evaluate that, one, the assay needs to be developed, and two, that assay needs to be compared to trapping to really determine which method is a better method for the detection of sirens. And so, you know, this half of the talk is really going to focus on sort of these two main objectives. One is the creation and validation of this eDNA assay, and sort of where we've tried to figure out the optimal ways of water collection and filtration, DNA extraction and amplification, and then you know, using those methods to confirm eDNA presence with both lab and field samples. But then as well, to compare these detection rates between eDNA surveys and trapping, and really figure out what's best if you're interested in detecting sirens on the landscape. So with eDNA, these methods have been sort of fine-tuned by us over the past few years, but really we're collecting this water from a site with a pitcher that's attached to a telescoping pole, and really that's the goal of that is to prevent disturbing the sediments and substrates from walking into the water and capturing those sediments in your water sample. We use a 47 millimeter filter cup that's attached to a PVC arm that you can see in this figure here, and that sits in a hand-powered fluid evacuator that's used in the auto industry. And so this is a pretty lightweight and portable sort of setup that allows us to filter these water samples in the field without us having to transport them back to the lab and filter at the lab. We use distilled water as a field blank, as a way to determine if we have contamination, and I'll discuss that a little further later. We take all our field samples in triplicate, and we shoot for roughly a liter of water passing through a Wattman grade 4 cellulose filter. And the pore sizes on these filters is 25 microns. And this is quite large compared to a lot of eDNA literature that's out there that use, you know, pore sizes as, you know, that are less than 1. And so it's really a trade-off between you know, balancing the amount of water that you can filter with these larger size pores versus you know, really capturing DNA with those tiny ones. And we find that we do still get detections with these larger pore size filters, and it allows us to still filter a large quantity of water. Once we've filtered water through a filter, they're placed in tubes with a sort of a buffer solution called DNazole. And then between sites, we clean all of our equipment with bleach and rinse it with sodium thiosulfate and and distilled water. And that's really just to prevent contamination and false positives between sites. So we don't want DNA to carry over from one site to another. Once we're back in the lab, we use a pretty standard chloroform DNA extraction with an ethanol precipitation to extract our DNA from the filters. And we have designed primers specific to Texas siren. So a lot of the mitochondrial sequencing that Rick Klein just talked about was used to help us tailor and modify our primers. And so we targeted a region of the cytochrome oxidase 1 mitochondrial genome as our target. And we tested these primers against several steps, at several iterations. One being siren tissue, where DNA concentrations are relatively high. We used field samples spiked with aquarium water, siren aquarium water, so that we would, hopefully get a positive from a site, but also have to deal with the various things that are in our field water samples. And we've also tested the primers against sympatric amphibians to really examine specificity of this primer set so that we're not amplifying DNA of non-target amphibians. And we ultimately settled on this, an initial plus a nested PCR round to amplify the small quantities of DNA that are typically captured with on these filters and from these eDNA samples. And, initially going in, our first round of PCR uses about 5 microliters of our eDNA extract. And then once that PCR is complete, we purify the product and then use 5 microliters of that purified product from the first step to then go into the nested round. And I'm happy to discuss more about the sort of nitty-gritty steps of this, but I've left that off the slides for now. And so when we've looked at our primer sets for specificity, we're able to get detections from sirens from multiple spots across Texas. So, with animals from Cameron County, Harris County out in Houston, and further west in Kinney County. And so we see detections there. We also get amplification from sirens from Arkansas, Mississippi, and Oklahoma. So there seems to be, this primer set in this assay seems to probably work with sirens from the greater Texas area. And it doesn't seem to amplify DNA from our sympatric amphibians. The sympatric amphibians we tested were South Texas amphibians, but as you can see in these gels, you don't see the positive bands would indicate that DNA is being amplified. So we're not getting detection of other amphibians from the region. So with a nested PCR, we sort of visualize our results kind of a couple different ways. One, we run the products out on a gel and look for bands of expected size. And so that nested PCR fragment is 178 base pairs. But to get better or more confidence in understanding that our DNA we're amplifying is truly sirens, we would also sequence the nested PCR product via Sanger sequencing. And we would get our sequences back from that and compare them with published siren DNA sequences that are on GenBank. And, usually when we did that, there's some existing data on siren genotypes there, and we're getting high percent match. And so we're feeling confident that the DNA we're amplifying and the bands we're seeing are truly siren detections. And so, so we've been able to sort of develop and create this eDNA protocol using this initial and nested PCR rounds, and which is shown to amplify and match sequences to sirens from numerous locations. So it's not just a South Texas specific thing, which we are fine with, but it seems to be still specific to sirens and not amplifying any sympatric species. And it does a good job of amplifying DNA from field samples. So the thing with eDNA is that your DNA concentrations are pretty low. So this nested PCR step really helps to amplify DNA to the point that we can quantify it better. And so we felt pretty good about this. And so we really wanted to put this to the test and see how how eDNA detection sirens compare to kind of the traditional way of trapping. And as Rick mentioned, so we did this study down here and we used these pretty basic minnow traps. So we sampled 5 sites in Cameron County, Texas, and those 5 are Resaca de la Palma State Park, the Texas Parks and Wildlife Olmito Fish Hatchery, which is now a coastal fisheries office, the UTRGV Brownsville campus, Resaca, the Sabal Palm Sanctuary, and the Nature Conservancy's Southmost Preserve. And so we would visit each of these 5 sites once a month, and we did this from March 2019 to February 2020. And we would collect eDNA as previously described and process it the same way using this nested PCR. At every site, every month, we would collect 1 blank. That would be our field blank of distilled water, and 3 eDNA replicates per water body per month. And again, we would target 1 liter of water passed through the filter, but we would also write down when we couldn't achieve that. So sometimes due to high turbidity, or if it had recent rain and there had been lots of runoff, the turbidity would be quite high, and those sediments would interfere and block and clog up the filter faster. And so sometimes we were not able to get 1 liter of water through our filters. And for it to be a positive detection of siren, we had this criteria of 2 of our 3 replicates needed to match published siren sequences of greater than 95% similarity. And we also needed no amplification in our blanks. And those blanks are both the field blank to sort of ensure that we're not getting false positives and contamination from previous sites, as well as our no template control, which is a lab standard to ensure there's not contamination once we're processing the samples back in the lab. For our trapping component of this work, we would set 3 minnow traps per sort of 6 locations within a site. So these locations are simply different spots along the margin of the resaca or the wetland that we're sampling. And so that resulted in a total of 18 traps per site per month. And again, they were baited with chicken liver and set in shallow water, usually amongst vegetation when possible. And we would set these traps overnight and check them in the morning and record sirens that were trapped. And so for our siren trapping results, so over a year of trapping at 5 sites down here in Cameron County, a total of 66 sirens were captured. Our most abundant site was Sabal Palm Sanctuary, where we trapped 34 individuals. And we were unable to trap individuals at the Olmito Fish Hatchery. And as a sort of side note, that existing work or previous work done by Rick Klein and his students, they've trapped at Olmito Fish Hatchery for 30 days, and every day they were unable to find any sirens except for the last day. On the last day, they trapped 1 individual. And so really it sort of is an anecdote that highlights the sort of unpredictable nature of trapping and that it can be difficult to detect these species. We know that there are sirens at this fish hatchery. In recent years, there's been anecdotal evidence of people seeing them, but there's also a large series of specimens collected there decades past in at the museum at UT Austin. And so we know it's a spot, but we just were unable to trap any individuals there. And as you can sort of look at the total trap results there at the bottom in this figure, we sort of see this more individuals trapped in the spring, kind of in April, again in another peak there in fall, and typically this probably corresponds with when there's water or abundant water and water temperatures and other seasonal effects of the species. So with our eDNA results, we had overall almost 98% of our samples, we had positive detections of sirens. And so unfortunately, we were unable to trap or collect eDNA at every site every month, and that's simply just because in the summer, sometimes the wetlands would be dry, so we could not collect an eDNA sample. But in total, instead of our 60 samples, which is what we aimed for, we had 46 eDNA samples, and 45 of those tested positive. And again, you can kind of see these are gel images at the bottom. You can see the eDNA, those 3 replicates producing a band there in that yellow box, but the field blank in blue and then no template control in red. There's no visible band there, which is suggesting that there's no contamination and we don't have false positives. Excuse me. And so to summarize the sort of this methods comparison that we've done, again, the comparison was one eDNA sample, which is 3 replicates, with 18 traps per site per month. And using some occupancy modeling, these probabilities of detections for trapping was about 0.45, and for eDNA, 0.97. So quite a stark difference there, and it is a significant difference in our detection rates between eDNA and trapping. So we're getting significantly higher detections with eDNA surveys than we do with trapping alone. Again, just reiterating that sort of stark difference in our probabilities of detection between our 2 methods. And so really, you know, this, in our opinions, validates eDNA as a very useful tool for studying siren distributions more effectively. We know that eDNA collection is considerably less effort than trapping alone. We really just need one visit to a site. You don't have to make repeat trips to check traps and place them and pull them. Typically, the permitting and restrictions are lessened just because you are collecting a water sample. And you can do this with pretty minimal disturbance to the habitat. Again, we're collecting that on a telescoping pole, so we're not even really getting into the water to disturb it. And, you know, once you're in the lab, you can sort of crank through your, you know, processing samples in sort of a high volume, high yield manner. And it's still important to know that sort of the siren biology and ecology is still understudied, and those sorts of questions cannot be answered from eDNA alone. And so we think that eDNA as a method can be used as a sort of a preliminary survey technique to reduce our trapping, sort of blindly trapping and really focusing on water bodies that we think should have sirens because of positive eDNA hits. And so we view this as an additive research tool and not a replacement for trapping. You know, we just simply can't answer questions about the ecology with much information based on just eDNA detection alone. And so with that, I guess we'll transition to some questions, and questions can both encompass what I've just talked about in the eDNA surveys, as well as some of the genetic work that Rick Klein talked about. And I think Paul will talk more about it, and he'll moderate the questions. But yeah.
Well, thank you very much, Rick and Drew. That was great. We do have a couple of questions in the chat here. The first one is related to the phylogenetics analysis, and the question is: did you use more than one outgroup for the analysis? Using only one Lacertina sample you could perhaps miss weird patterns such as polyphyly.
Okay, I can answer that. Yes, for the phylogeny that I showed, I'm using one outgroup, but we have used Pseudobranchus. We have used other frogs and other species for the outgroups, and we get the same pattern. You can also see in that 2018 publication regarding Siren reticulata, we use several outgroups. The problem becomes that there's lots of variation, even in the mitochondrial sequences, and I guess the coverage that we get from some of the other sequences published in GenBank don't include some of the things that we sequenced. So that's why in this particular case, we used a Siren Lacertina that we sequenced as well.
Thanks, Rick. Nate actually followed up. He said he specifically meant more Lacertina examples. I think that was my bad in reading the question. Since they're reported to be in South Texas as well, so he'd be curious to see if more extensive Lacertina coverage?
Oh, certainly. I mean, I have, I think, 3 Lacertina complete mitochondrial genomes that we can use. And then there are quite a few partial sequences in GenBank that have 16S and CO1. And I have a very large phylogeny that we made using all available sequences from those. And that's where we get the idea that, I guess, Lacertina and their intermedia that's been described and published in GenBank all kind of grouped together well away from anything that we capture in Texas so far.
Thank you, Rick. Okay, there's another question here. Did the Siren genetic work have enough known males and females to look at sex-specific markers?
As of yet, we know of no external means to sex these sirens. We were taking tail clips. I don't think there's enough known about the reproduction of these sirens to say we certainly don't know.
Okay, great. So another question. From past work with greater siren in South Carolina, we've found that it is important to use a variety of trap types because of the size-specific biases of each trap type. We found that steel minnow traps typically captured smaller animals versus trash can traps or fyke nets. Have you noticed any size-specific biases in trap result comparisons of crayfish versus minnow traps?
In the beginning, I guess, Drew, you want to?
Well, yeah, I mean, I can answer that. We use minnow traps, but we did expand them, the sort of the funnel to make them slightly larger. We have, usually the crayfish style of those metal minnow traps have a larger opening, but certainly there's a lot more variation in it that, maybe Rick can comment on it if they've done some previous work down here. But we have been sticking to these metal minnow traps, and it's hard to know if we're selecting against truly large, giant animals. But yeah, maybe Rick can elaborate a little bit more.
Yeah, over the years we've used a number of different baits and trap sizes. I think the problem becomes we needed samples to look at, and sometimes you catch nothing at all. As Drew mentioned, we trapped at one area 30 days in a row, and only on the last day did we capture 1 siren. There are probably bait preferences for different species, and trap size limitations, but I guess we would expect that we would at least catch a juvenile if they were, abundant in the area with the trap sizes we use. The minnow traps and the crayfish traps we use, I mean, we can catch an animal that will have its body sticking out the end of the trap. It's so large. Like I said, the largest one that we captured, I think, was 600 millimeters, and the smallest one was a very, very small juvenile. So there's probably some more work to be done there in those comparisons, but I guess in comparison to other areas of the country where you can go out and trap all the time, in South Texas, we get a lot of times a year when there's no water or so little water, the water's so hot that you catch nothing. So it's kind of a trade-off in terms of that.
All right, thank you. There's a kind of a comment and a question here. Siren were once very abundant at Olmito Hatchery. Any idea what happened there?
Yeah, I have no idea. Yeah, there's a large series that are represented in museum collections, and we know they're still there in recent years. We were unable to trap them over the 12 months of sampling. despite there being consistent, it was a site that never dried out, unlike some of our other sites that are like Sabal Palm Sanctuary or the TNC Southmost Preserve. Those Risacas would dry typically in the summer, and then just depending on the land management, they did buy water to fill them back up. But really isn't how that Risaca is managed at the Olmito Fish Hatchery. I do think that there could be a different community if more fish are present there. We didn't really have direct evidence of that. We know that they have been seen there in recent times, just anecdotally by some of the workers where they've, I think one of them pulled back erosion netting that had blown in, and a siren was in and amongst that. But yeah, we certainly get their eDNA there, but we're not able to trap them. And so it sort of is a curious situation. The fish hatchery does do, again, it's a coastal fisheries office now, and so they do a lot of work with fishes in the Gulf, but they come back where they collect them and do, put their scrap in the water. And so it could be that they're feeding on different things or just not attracted to our bait for whatever reason. Don't really know.
Okay, thanks, Drew. Another question here. What's next for establishing the range of the siren? Some sampling in Mexico? And, I'll add to that. Were you able to include any tissue samples from Mexico in any of the work you've done so far?
That's been a goal of ours forever. One goal was to get this Maverick County sample that kept floating around, and I finally got a hold of it. And by the time I did 2 years later, the tissue was so degraded I couldn't get any sequences out of it. There was a sample from Mexico in the museum collections, and it has been processed and used. And with the violence going on down in Mexico, I know specific locations where we could readily capture them. We just have not been able to get down there to get them. I wish I could find a collaborator that could help me out with that, because I think it'd be very interesting to see if there were different genotypes down there. But so far, no luck with the Mexican sirens. Funny thing is that they'll post them on the similar website to iNaturalist, and you'll see people holding them in their hands in Mexico. But try getting a sample up here is another thing entirely.
Okay, thanks. Another question. What do they eat?
Yeah, I guess we don't specifically know that in the sense that we haven't directly, flushed stomachs or examined things. Sometimes when we are holding sirens for processing and sampling, they often defecate large numbers of snails. But again, snails have that hard shell, and so there's evidence of that, of course, in the feces. Other things, of course, are lost. All the soft-bodied invertebrates that they're probably eating as well are too digested, and it's hard to tell from fecal samples. But it hasn't been something that we have examined in recent years directly down here.
Okay, thanks. Another question. Do you guys have any theories regarding how a separate species would have evolved in South Texas and northern Mexico, given that their distribution is or very nearly overlapping with Intermedia?
Yeah, I do not. I think we have a little bit of preliminary evidence with the black-spotted newt showing that maybe there was some effects of glaciation that happened long ago that could have pushed animals down further into Mexico, and then they repopulated themselves back up this way. And I don't know. I guess the siren distribution actually ranges all the way up through apparently Michigan, so they can probably withstand some pretty cold weather, but I don't know for how long. That would be one idea, but they live in the same habitats. They seem to behave the same. I have no idea how they recognize their mates from other sirens, so that would be something interesting to study.
and I'll add on to that a little bit. Yeah, historically, the reason I think partially attributing to this is that the perception was that these sirens are larger in South Texas, and it's certainly a different landscape down here than much of sort of the southeastern United States, which is a lot more mesic, a lot wetter, a lot more rainfall. And so maybe there's, you know, certainly different selection pressures for these individuals down here for estivation and, you know, having these habitats that dry regularly. And so, you know, the sort of maybe mindset would be that larger individuals can estivate for longer and really hunker down between these dry periods, between these hurricane rains and things like that. That probably that's required. So.
And I have one other thing to add there. I guess Texas had an inland sea that divided it for a while near where the Pecos is now, and it could be that they were prevented from crossing that barrier for long enough. Although we haven't found any evidence of that being the case yet for a different species of siren.
Okay, great. Okay, another question. Do you expect the Rio Grande to be a significant barrier to dispersal, or would they just swim right across it?
The Rio Grande, is that what you asked?
Yeah, if it's a barrier to dispersal.
I don't believe it is. I mean, we have tested water from the Rio Grande, and we do get positive detections of sirens there. It wasn't part of our trap study comparison work, but anecdotally we do have evidence of their DNA there. I wouldn't view it as it. I mean, it can be pretty shallow, and I see no problem with, especially if it was high flow and there's water and suitable habitat on both sides.
Okay, another question. I've heard about sirens being able to vocalize. Did you ever hear anything like this during your trapping experience?
I guess I can answer this one. So yeah, they do vocalize. They make a little chirping noise almost when you're handling them. And I think it's just air in the buccal cavity making that noise, but it is kind of cute. We've often wondered why exactly they're called sirens, but that may be it. Drew, I don't know if you have anything to add about that, Drew, but all I know.
No, yeah, I haven't experienced it, but I certainly, they make noises when they come up and gasp for air or breathe at the surface, and but yeah, I haven't had a lot of experience with it.
Okay, there's a comment here. It says they were called sirens by Linnaeus because of their front half is terrestrial and their back half is aquatic. It's very interesting. Okay, well, that's about all of the questions I can see. I think I've got them all. So in that case, we'll go ahead and wrap up. We're a couple of minutes away from being done. Drew, if you wouldn't mind pulling up that other slide real quick. I just want to say thanks again to Rick Klein and Drew Davis for their work here and for giving us a good talk. And as a reminder, this is a series, so we have talks every month, the third Wednesday at noon. The next talk up on deck is the status of the Swift Fox in Texas up on September 16th. So we'll be sending out a notification for pre-registration and all that at some point in the next 3 weeks, 2 to 3 weeks. And we have a series of other talks coming up for the rest of the year, a nice variety of stuff. So thank you again for everybody's attention, and yeah, look forward to seeing you again soon. Thank you.